Greetings, everyone, and welcome to the Pan Future Podcast. Today is Saturday, February 24th, 2018. I've got an interview today coming up with Dr. Kevin Folta. It's the second time on the show. We get to talk all about plant genetics. But before we get to that, let's get to some news of the moment. In the news this week, the clock of the long now is officially under construction. I have talked about this several times before. Talked about the Long Now Foundation and the clock itself. It is a clock that will run for 10,000 years. It's being built on some property in Texas owned by Amazon founder Jeff Bezos, and he is also one of the main backers of the Long Now Foundation, along with some other celebrity-level people. And this has been in the planning phase for quite a long time, and it's really exciting for me to see this finally begin to undergo its actual construction. It's really a marvelous, amazing piece of mechanical design. And just the psychological, philosophical, and cultural prompting for thought that this can bring into the world is remarkable to me. As I have talked about the clock before, this was one of the things that really prompted me to do this show, to think about long-term thinking, truly long-term thinking. 10,000 years. Just think about that. That is longer than the, what would you call it, human civilization in general, especially anything that's been written down, ancient Egypt or Babylon. 10,000 years is longer than that. Boy, I hope we can stick around that long. And it is unlikely that anyone listening to the show will actually live that long. But let it be a prompt for you to consider when you're making decisions about your life, what you do, how you interact with the world, how you treat other people, the kinds of decisions you make at the voting booth. It matters not just what happens tomorrow or next year or the next election cycle, but the choices we make now have an impact on what happens 100 years from now, 500 years, 1,000 years from now. And for me, this clock is just kind of a beacon to stay focused on for that long-term viewpoint. All right, that's the bit of news I wanted to hit you up with this week. Now let's get to our interview. All right, this week on the show, I would like to welcome back Dr. Kevin Folta. Kevin, welcome back to the show.
Hey, yeah, really nice to be back. Thank you.
Yeah, thanks for setting aside some time. So this week we're gonna focus in about plant genetics and improving those genetics. Where should we start with that?
Well, I think the best place to start is let me ask you some questions. Okay. I'll flip this on you.
All right.
Okay. What are some things that you worry about, or that you think that most people worry about food and farming?
I guess high on the list of fears and misconceptions is that my tomatoes have fish genes, or my potatoes have genes from something. The Frankenstein fear, I guess we could call it. You mash two things together into some sort of weird chimera, and it's going to be unleashed on the world.
That's interesting, because we'll talk about that, and what are the more prevalent chimeras, like as you say. What are the things that are really mashed together when we genetically improve plants?
Okay.
And it's something that I always refer to as the Frankenfood paradox. And what it means is that the least random, least risky methods that we use to improve plants are the ones that people fear the most. And the ones that are random and crazy are ones that get a free pass.
So what people feel comfortable with the ways we've crossbred plants and other things over the years. Obviously, your average farm cow or pig or chicken is certainly not like its wild relatives either. When that topic comes up, I often like to refer to the Africanized honeybee as a case of unintended consequences. So what makes the specific genetic engineering of single genes a better method?
Well, let's start by talking about the idea of plant domestication. It's one of my favorite topics. Mm-hmm. All of the plants that we consume are nothing like what was there in the wild. As you mentioned with animals during animal domestication, the plants that grow in the wild aren't even from North America, for the most part. Mm-hmm. Citrus comes from Southeast Asia. Peanut and tomato and potato come from South America. Mm. Corn, squash, beans from southern Mexico and into Central America.
Yeah, one of my recent favorite pictures I saw on the internet was one describing the ancestor plant for broccoli and cauliflower and kale and Brussels sprouts and something else that all comes from the same plant.
That's right. Yeah, it's Brassica oleracea. They're all interfertile because they're all the same species, but they all have different looks because of different traits that humans selected. Mm-hmm. So in a way, the broccoli, cauliflower, kale, kohlrabi, and cabbage, and Brussels sprouts are analogous to Chihuahuas, Great Danes, Boxers, Great Pyrenees, Shih Tzus, and Bulldogs. They're all interfertile, and they can mate. Yet humans have selected for very specific traits that they continue to select for because they find them desirable.
Now, with modern genetic engineering methods, instead of crossbreeding a plant for certain traits over many generations, we can shortcut that process quite a bit, can't we?
Yes, we can. And that's what's so exciting, is that the process of breeding, of facilitating a human presence in plant sex, taking pollen from one and putting it onto the stigmatic surfaces or the female surfaces of the other. That whole process is something humans have been involved in to genetically improve plants that hasn't changed much until the last 50 years. We've just stepped in finding something we really like that has some good characteristics, and then find something else that has good characteristics we like, and then try to get all of those characteristics in the one genetic background. That's breeding and selection. Mm-hmm. We've been doing that for a while, probably 20,000 years or so, with some rather ancient crops. Things like strawberry maybe only the last couple hundred, blueberries the last 100 years. So genetic improvement just by being facilitators of plant sex is a relatively new phenomenon.
Mm-hmm. And in the case of selecting a very specific gene to insert into a plant, how long have we been doing that? And that's just another step on the same road toward getting the exact traits we want, right?
Right. Well, what it means is that when we're breeding plants and doing the plant sex thing, you're mixing tens of thousands of genes that you don't understand with 10,000 genes you don't understand and mixing them together. And all the different variations, all the different variants, all the viruses that move along in the genome, all the things that you're mixing, you have no idea. You just know that you get something that you like out the other side. Mm-hmm. When you're genetically engineering something, you're moving one gene that you understand in tremendous detail. And you're moving it maybe from plant to plant, maybe from a bacterium to a plant. There are no examples where you move a fish gene into a tomato. I mean, that was a hypothetical, and actually it was something that was tried. It was trying to put an antifreeze gene from flounder into some plants, but it's never been commercialized. Mm-hmm. So the idea is that, and this is part of the Frankenfood paradox, the genes that we understand extremely well and the processes that we understand extremely well are the least risky and best understood and the most feared. It's very interesting.
It's the least risky because you're you're handling one specific gene and you know what it does, you know what to expect, rather than the randomized way that even if we facilitate the breeding, it's still kind of like it is in the wild. There's a lot of variations and chance that goes into it.
Exactly. We know exactly where that gene lands. We can tell you where, if it's disrupted other genes. We can tell you if it's changed the expression of other genes. That's stuff that we can do when we genetically engineer something. And you mentioned before, how long have we been doing this? It has been happening in bacteria since the 1970s and has been done in plants since the early 1980s. So this is, at this point, a 38-year-old or 36-year-old technology that is really very, very mature.
Yeah. So, yeah, I guess I should mention the opponents of genetic engineering often like to turn to the tomato or the whatever full of hypodermic needles, as if that's how this is actually done, and I'm quite sure it's not. So can you describe the actual process that you go through, the technical bits of it?
Yeah, these folks are really good at finding the tomato junkies, getting them still. Really, what it boils down to is it's a couple of different methods. But the first thing to point out is, if that's the kind of rhetoric people are using to dissuade people from science, it really shows how dishonest they are. That trying to create this fearful, dreadful, everybody dreads getting a shot, right? And creating the dread and the fear around technology, which I think is really unfair, and it hurts the poor the most. How it's really done is actually a much more sophisticated process, and really boils down to a bacterium called Agrobacterium tumefaciens. So Agrobacterium is a bacterium that lives in the soil, and its natural life cycle causes it to infect plants by installing some of its DNA in the plant. So what the agrobacterium does is a little piece of DNA in a specific part that it transfers into the plant cell. And when that piece goes in, it gives the cell new instructions to divide prolifically and grow into a big lump of cells. So when you see these things on trees that they call galls, like these big tumors, that's from an agrobacterium infection. So what scientists managed to figure out was how agrobacterium did it. How did they move that piece of DNA? Then what they did was they disarmed that bacteria, took out all of the genes that lead to the cell division and tumor formation in the plant. And it's not really a tumor, it looks like it. It's actually called callus. It's just bubbly tissue, differentiated cells. It took out the genes that caused that, and they added the genes that they wanted to add. So if you wanted to add a gene to a plant to make it survive herbicide, or a gene that might be toxic to an insect, then you could add that gene to agrobacterium in that one little part of its genome, and then it would transfer that to the plant material. So you're using this natural process just to do the work of moving the gene to the new organism.
This is something that's the bacteria occurs in nature. It's always been doing this. You're just adapting it to a different purpose. Maybe a metaphor that wants to pop into my brain is sort of like, you're riding the bus, and the bacteria is the bus, and you're just selecting which passengers are on the bus going to a destination.
Yeah, it's pretty true, and it's a pretty good analogy. You're basically, the scientists are putting the genes they want into that payload, and then the bacterium just goes and delivers them, drops them right off at the station.
Is this, when the bacteria does this, does it propagate itself, or is it kind of a one and done thing? Maybe one of the fears that some people have is something will spread into the wild or into non-GE crops or something like that.
No, that's an excellent question, and let me answer this in two ways. The first thing I can tell you is it is kind of a one and done thing. The bacterium makes that exchange into one cell. And one cell is not a plant, right? I mean, one cell is just one cell, but it has all the information in it to make a plant. And just like when you can cut a cutting off of a coleus or a basil plant and stick it in water, and those cells on the stem realize they're not in the right place, and they start to grow roots, start to turn into roots. Plant cells have that plasticity. You can be a root cell or a stem cell or whatever. So when you get that gene into that one cell, and now you put that cell on the right laboratory medium, the right type of agar, the right plate of goo and nutrients, then that one cell turns into 2 cells, then it turns into 10 cells, turns into thousands of cells, and eventually may turn into, start to realize that it has the identity of a plant. And it might start to make leaves and roots. And pretty soon, all of those leaves and roots that came from that one cell now have that new gene that you've installed. So that's the whole idea of getting to the plant. Right. Now, to your question about, how can it spread? It could always cross with anything sexually compatible. So if you had genetically engineered corn in the field, it could pass on, potentially, that genetically engineered gene to other corn. Sure could happen.
Off the top of my head, I don't really know what the fear of that is, other than that it's somehow tied into the mistaken belief that Monsanto sues people who get accidentally crossbred. There's not really if you're creating something that's beneficial for the growth of the plant, you're not going to accidentally create some sort of blight that kills a whole bunch of other things. I mean, there's not really a connection between those two.
Well, it's highly unlikely, because if you had something in that first line that was deleterious, it would kill the first line. All we're talking about is a gene here. We're talking about some information. And if a plant was developed that had the potential to kill other plants, the first plant that it was in wouldn't live. People forget that plants are really sensitive organisms to their environment and to their surroundings, to their nutrition, to water. And if you were to put something into, people are worried about eating stuff from plants or eating the plant material themselves. But think about the plant. It's living, perfectly fine. Its cells are very much like ours in many ways. Of course, different in a lot of ways too. But the idea is that the plant cell can tolerate, these disruptions because they're favorable.
Maybe we could get to some specifics too. Am I right in remembering that you work with strawberries?
Yeah, well, we do a lot of work with strawberry with the idea of genetic improvement for flavor and aroma.
Okay.
And that's why that's important, is that we don't do it with genetic engineering. We come up with understanding the genes that are involved in secondary metabolism, and then help our breeders by developing ways to test a plant to see if it has the favorable genes. That's our job.
So you're talking about all the genes that are already in the strawberry. You're examining ways to maximize which ones are on and off to help improve the flavor. Or do you also improve the durability of the fruit?
Well, it's possible to do all those things. I think what we do is we try to identify, and it's not just on or off, it's also present or absent. Maybe the plant doesn't even have that variant of the gene that's required for the plant to make the product. So what we do is we have diagnostic DNA tests, almost like a paternity test, where we can ask, does that plant have the hardware to make the good tasting chemicals? And if it does, then our breeder may choose that plant in his crosses.
And do you get pushback in any way with this specific kind of work of examining the genetics of a strawberry and deciding which plants are the most favorable? Does that go too far into it's not really even frankenfood, it's more like just really understanding what's there? But do people oppose even that?
Well, there are people who say, I want food just as it appears in nature, which is unproductive and awful. I mean, humans have genetically approved plants.
Yeah.
And so some people do push back. But in general, people don't mind traditional breeding. They don't mind the examples of, emasculating a flower, which means ripping out all of its male parts. Be careful if you Google that, by the way. We have a strawberry line in Florida called Sweet Charlie, and it's a variety. And I put in once emasculation and Sweet Charlie, and what came up in Google Images was not for the weak of heart. But you tear off the male parts, and then you have the pistillate, or the female parts of the flower, and now you take the pollen from the male parent, which might be a completely different genetic background, and add that to that pistil, and then give it a little covering so it can be nice and moist and do its thing. And what's really nice about that is that This, we now just made a genetic combination that never existed in the history of the world, that may or may not have very favorable characteristics in the next generation.
Mm-hmm. When you're growing plants that you've made with this process, how do you get from the lab samples to the farm field with tens of thousands of plants?
Yeah, that's a good point. Well, nowadays it's pretty easy. We have expertise, and this isn't my lab. This is our strawberry breeder, Vance Whitaker. Vance is very adept at making the right crosses. He then screens at least 10,000 plants in the field. And traditionally it was done this way. You'd screen 10,000 plants, find maybe 200 that were good, just by walking the rows and looking for things like fruit size, color, yield, disease resistance. You'd look for those kind of characteristics. Mm-hmm. And notice you never looked for flavor.
Right.
You first look for the things you could observe. And then if you had that, the next year you might take those 200 plants, make 20 of each one, and then check them again and see if you still get good characteristics, and then maybe start looking at flavors. So that's the way it was done traditionally. And maybe after 4 or 5 years, you would be down to one plant that was the next new variety. Mm-hmm. What we do now is we know the genes that are favorable for specific disease resistance and taste and other characteristics. And Vance does this. And they screen 384 plants at a time in a Petri dish and can tell you from the seedling which plants have the best stack of traits. And what's so cool about that is that you don't have to plant 10 acres of plants, which requires fuel, labor, plastic, fumigation. Mm-hmm. Now you're able to do all of this in a Petri dish, and it's much, much, much more powerful. So now the 10,000 plants that make it to the field, if it's even that much, it might be 1,000. We know that every one of those plants has an outstanding potential to be the next big cultivar to help farmers remain competitive and give better fruits and vegetables to consumers.
Mm-hmm. Does this process work with pretty much any plant? Do you see this kind of thing going on with corn, soybeans? I'm also thinking about fruit trees and nut trees might be difficult if you have to grow a whole tree.
Sure. Well, you're exactly right. I mean, this has been happening, and what this is called, the term for this is marker-assisted breeding or marker-assisted selection. What it means is we're able to look for specific signposts in DNA that tell us that the gene variants that are useful are present or absent, whatever. What's nice about this is that it does accelerate something like tree breeding. Can you imagine growing an apple tree, making a cross, because you really like this one and this one, so you put them together. And then you're growing those seedlings 3, 4, 5 years before they flower and give you fruit. Mm-hmm. It's a slow process. Yeah. So having some markers that help you pick the best seedlings and the ones most likely to have the best traits, that is a major step forward in how we genetically improve plants.
Mm-hmm. I hope this is giving people a better insight into what people are actually doing. Part of me really struggles to understand why people are afraid of this, other than that they have been marketed information with a fear-based campaign, which really kind of makes it no different than the way the anti-vax people are, or the way a lot of our modern politics is based on fear. What would you like to give us a closing if people hear about technologies being used in modern farming? If they are concerned, what would you say to them?
I would say the same thing I say about every situation in life. You never make a decision based on fear. And that's a scary thing to say. You have to have the courage to trust science. Mm-hmm. And that sounds ridiculous. We do it with medicine all the time. We're not so sure about food. And it's mostly because of the tremendous disinformation campaigns that have been extremely effective in polarizing opinions towards food. So my feeling, learn what you can. Surround yourself with good information. Double check it, triple check it. Look at all of the activist information. They have the same concerns we do. They're worried about the environment. They're worried about human health. We're on the same page. The thing is that they sometimes have some bias in mission to defeat good technology, where my job is to promote the best technologies and develop the best technologies. So learn from everybody, learn who you can trust, pay attention to your university experts, and see how, most of all, does the information come from peer-reviewed research? And if it does, have other people repeated it or expanded on it. And what you find is that all of the fearful messages are dead ends. They publish the paper and then it's gone. They're always standalone papers. And when people say, well, look at this paper from 1997 when they fed potatoes to mice. Well, yeah, but it's 2017. It's 20 years ago.
Yeah.
That's 2018. That zombie paper has had a stick in it for a long time.
Yeah. Do you have anything to say about some of the information people get? Some of the things that people have thrown at me as citations are like WHO reports and what is it? The Environmental Working Group, which to me is a bit sketchy, or more than a bit sketchy, but it appears to be legitimate sources of information. Or, like the WHO should know what it's doing, and yet sometimes seems to clearly get it wrong.
Well, we won't get fooled again. That's my WHO joke. No, actually, so what you're referring to is the IARC, which is the cancer evaluation arm of one part of the WHO.
Okay.
World Health Organization is an outstanding organization to follow for consistent good messaging. And independent arms of the WHO do not endorse the IARC finding with respect to, and what the controversy is an herbicide used in genetically engineered crops, glyphosate, saying that it is a probable carcinogen, when the evidence doesn't really say that when you objectively analyze it. There's nothing that suggests that. So then the other thing that we're dealing with is the other good sources of information. You mentioned another one. What was it? The WHO?
The Environmental Working Group comes up a lot. Yeah.
Environmental Working Group. So what an environmental working group does is they use legitimate USDA statistics on detection of pesticides, because food is monitored for pesticide residues. It's important. Our environment is monitored for pesticide residues. It's very important. And they publish their findings. And what they find is parts per billion, parts per trillion of different pesticides. And these are way below, hundreds or thousands or tens of thousands of times below any thresholds of human relevance. What EWG will do is they'll say, well, we looked at strawberries and we found residues of 52 different pesticides. Now, a strawberry farmer out here in Florida might apply 2 or 3 during a season, and apply 2 or 3, and then the field has to be, evacuated for a certain amount of time. And then when that interval is over, you can safely touch and you can safely eat them right out of the field. No problem.
Yeah.
But what EWG does is they say, we found on strawberries 52 different pesticides. You want your kids eating 52 different pesticides? EWG pushes families away from produce. They push families away from healthy eating, and in their agenda, they actually make things worse.
So it really seems like they're just taking things way out of context. Sort of just they're throwing out that fear headline, and without any context around it. I think one of the responses to that I saw was that I think it was to one of the pesticide concerns that somebody had tossed out, was that the risk, the carcinogenic risk, was the equivalent of drinking 1 glass of wine every 7 years.
Yeah.
So it's all this nuance and gray area and detail that everybody loves to skip over. I'd really like to see the media get its act together.
Yeah, well, the most risky thing you will do today is drive a car.
Yeah.
And if you had to put money down on somebody buying the farm today, it would most likely be in an automobile accident, because that's the riskiest thing we do. Yet we're pretty mundane about that. It's not a big deal to us to get into a vehicle. But my favorite one was the glyphosate in wine claim that there were parts per billion. So this is like seconds in 32 years, right? Parts per billion.
Okay.
And they're saying, oh, glyphosate, a probable human carcinogen is in parts per billion. But what about alcohol? A known human carcinogen is present in 13 million parts per billion. 13%, right?
Yeah.
So the risk perception is so screwed up here.
Yeah.
And that's part of the problem.
And really, it seems like just about anything could be carcinogenic in the right situation.
Well, yeah. I mean, I don't know that the IARC has ever found anything to be non-carcinogenic, or I think maybe one thing in all the stuff they've evaluated. Mm-hmm. And but they evaluate things like things that have been described as putative or probable carcinogens, pickled vegetables, night shift work, sunlight is a known carcinogen. So. And the state of California has at the border must have a big sign that says, sunshine in the state is described by the IARC as a class 1 carcinogen. I'm serious. This is how ridiculous this kind of context-free, hazard-based. So what they say is that basically sharks are dangerous, and so you're probably going to be killed by a shark today. Well, not if I'm standing in the middle of Milwaukee, Wisconsin. They identify hazards. They don't adequately articulate risk, which is something very different.
Yeah, which is too bad, because I think there's plenty of things worth worrying about in the world today, and I think most people aren't very good at figuring out which things are really concerning and which ones aren't.
Alison Van Eenenum, Dr. Van Eenenum says, and I can't remember the exact quotation, but it's in Food Evolution, which everybody has to check out. It's on Hulu, it's on iTunes or whatever, not on Netflix. So Netflix won't play a pro-science film. Which
was very disappointing. Netflix they have a reputation.
Yeah, you can see Food Inc. there all day.
Yeah.
But Allison says the risks that concern us and the risks that kill us are two very different things. And I agree with her. Humans are have very skewed perceptions of risk, especially as it applies to things like families and children. Yeah.
That's probably a good spot to wrap up. People out there, don't worry too much about everything. Figure out what you can really have some control over, and eat your strawberries. They're fine.
Celebrate your food, right?
Yeah.
Celebrate the fact that we spend less money of our income on food than anyone else in human history. Worldwide poverty is at an all-time low. Technology, farming, production techniques, what we're learning from organic production, all these things are really important. And we should be celebrating food, finding ways to mitigate waste, mitigate its carbon footprint, enact fair labor practices. Yeah. Those are the places that we should be focusing, not on sticking syringes in tomatoes. Yeah. And I think if we can do that, we'll be better off.
All right. Well, thanks again for your time, Kevin. I really appreciate it.
Well, my pleasure. Let me throw in where I can be contacted.
Yeah, you bet.
I host the Talking Biotech podcast every week. And also I'm at Kevin Folta on Twitter. And reach out. You can find my email anywhere online. The activists easily do. If you need anything, have some questions, send me a note. I answer every single one. Thank you.
All right. Thanks again, and take care.
Yeah, thanks so much. We'll talk to you soon.
All right. All right, that is your show for this week. Many thanks again for Dr. Kevin Folta for joining me on the show. And thanks so much to you for tuning in, listening, thinking about these things with me. I hope you've learned something new this week. I guess that's all I got. Everybody have a good week. Go to panfuture.org, find me on Twitter. Check out the show notes for some links and some videos. Go find Kevin on Twitter ask him some questions about plant genetics, and be better tomorrow than you were today. All right. I will talk to you again in the future.